Hello, my name is Dr. Badur Sahi. I'm here from the Translational Genomics Research Institute, where I'm an assistant professor interested in breast cancer and multiple myeloma research. I studied at the University of Toronto in cancer and cell biology, and I moved to TGen to explore new avenues of research. Today I'm going to talk to you about the basics on cancer and how we conduct research. I'll give you some basic cancer facts. I'll discuss a little bit of basic genetics. I'll explain what omics mean. I'll discuss the technology that we use in cancer research and a little bit about how we treat cancer. I'll explain what translational research is, and I'll discuss a little bit on clinical trials. Cancer is the second leading cause of deaths in the United States. There are different types of cancer with different incidences and mortalities, both in men and women. Breast is the most common cancer in women, and prostate is the most common cancer in men, and lung cancer is the #1 cancer killer in the United States. There are different types of cancer. There are over 100 different types, which treatments is based on the kind of cancer that's involved. Cancers are divided into 5 main subgroups. You can refer to cancers as carcinomas, which are cancers that begin with skin or tissues that line the internal organs. There are sarcomas, which are cancers that start in bone, fat, muscle, joint, nerve, blood vessels, or deep skin tissues. Lymphomas are cancers that start in lymph nodes or lymphoid tissues. Leukemias are cancers of white blood cells. Myelomas are cancers that start in plasma cells, which is found in the bone marrow. So what is cancer? Cancer is a group of many related diseases when new unwanted cells form and when old cells do not die when they should. These extra cells form a mass of tissue called a growth or a tumor. Not all tumors are cancerous. Tumors can be benign or malignant. Now, I was saying that cells grow when they shouldn't grow, and that is basically due to an imbalance of genes that control cell growth. So there are tumor suppressor genes, and there are genes that are cancer-causing, and the balance in cancers is shifted on the side on the tumor-forming genes. Cancer is an accumulation of multiple genetic alterations. Mutations can start that can inactivate tumor suppressor genes, which prevent tumor cells, normal cells, from becoming cancerous. Cells will then divide and progress along a path that can lead then to tissue invasion and metastases to other parts of the body. So when a cancer cell leaves its original organ and goes to another organ, another site in the body, it's referred to as metastasis. There are many hallmarks of cancer that we like to understand and study when we do our research. So the main hallmarks of cancer basically are these listed on the slide. So we have self-sufficiency and growth signals. Cells can just grow in the absence of stimulation. They evade apoptosis. Our body has mechanisms to eliminate cells that don't behave well in the body, and cancer avoids that. So you have the perpetuation of cells that are altered and mutated and malfunctioning, and those can lead to cancerous growth. You also have a sustained angiogenesis, which is essentially this means is that cancer cell can supply its own food supply, so it nurtures itself. It can grow indefinitely, and it can also, another major hallmark of cancer is that it can invade and metastasize to other organs. So I keep talking about a cell, and I just want to put that into a hierarchy for you. So what we have is that cells form tissues, which form organs and organ systems, and then full organisms. And the central dogma of molecular biology is very important to what we do in cancer research, because in the cells is what we want to study how these cancer cells form. And to do that, we need to look at the heart of these cells, which lives the DNA, which is our human fingerprint. And so DNA, the central dogma basically means that DNA becomes RNA, which becomes protein, which is the functional unit of the cell. And when these are altered as well, we have all kinds of problems that can happen. So DNA is the molecule of life, and it's estimated that there are trillions of cells in the body that carry a nucleus which lives the DNA. There are about 300 billion subunits that make up the DNA, And these subunits basically form words that transcribe into genes. These genes and this DNA gets basically accumulated and organized in a way that leads to chromosomes, and there are 46 in the human cell. And if you take the DNA and stretch it from one end to the other, it measures 2 meters long, and yet it can fit into a very microscopic part of the cell. The 3 billion bases of DNA make up about 30,000 genes that code for proteins that perform the life functions in the cell. And this is important and has a lot of implications to the Human Genome Project, which was completed in 2003. It was a 13-year project coordinated by the U.S. Department of Energy and the National Institute of Health. And during the early years of the Human Genome Project, many other countries became involved, and major partners all helped to basically make this happen. And one of the main purposes of the Human Genome Project was to identify the approximately 25,000 genes in human DNA. This number actually varies, so we see 25,000 genes to 30,000 genes, which is probably more the accurate representation. But it was also determined the sequence of the 3 billion chemical base pairs that make up human DNA. And since the end of the Human Genome Project in 2003, genomics has become the mainstay of biomedical research, because it allowed us, we knew the code in order to study the DNA. There have been numerous genomic achievements since the Human Genome Project, and this is just a pictorial demonstration of some of these achievements, and I have a reference here for you if you're interested in reading further about some of these achievements that were a direct result of the Human Genome Project. There have been numerous stages of accomplishments across, since the Human Genome Project, and this is just a schematic representation showing where we are now. We are at the point where we're trying to improve the effectiveness of healthcare, whereas before we were really trying to understand the structure of the genome. We have a pretty good understanding of that now, and we just keep shifting our focus to the various other aspects of the genome, of genome science. And now we're really, really focused on improving effectiveness in healthcare because of genomics research. So what does omics mean? It informally refers to a field of study in biology such as genomics, metabolomics, proteomics. And the related suffix "ome" is used to address the objects of study of such fields, such as the genome or proteome. So this was a word that was used in the context of the word biome, and later genome became a popular word for the complete genetic makeup of an organism. So I wanted to also discuss and just clarify the differences between the word genomics and genetics, which are similar and related, but not exactly the same thing. So as a rule, genetics is the study of a single gene in isolation, and genomics is the study of all the genes in the genome and the interactions among them and their environment. So I have two analogies to help you understand this a little better. If genomics is like a garden, genetics is like a single plant. The plant isn't flowering. You could study the plant itself, which is genetics, or look at the surroundings to see if it's too crowded or shady, which would be the genomics aspect. And both approaches are probably needed to find out how to make your plant blossom. So we do genomics research, but we definitely still also do genetic research, if that makes sense. The second analogy is if genomics is like an extended family, genetics is like one family member. A girl can study her romantic partner in detail and think she knows all the quirks, but she may have her eyes opened even more after seeing her partner behave at a family reunion. So that helps gives you sort of just an analogy to how we differ between genomics and genetics. So if we go back to the central dogma of molecular biology in terms of genomics, all the genes in our body, which is the study of genomics, leads us to about a study of 25,000 genes. The DNA, which then makes the RNA, which is the message of the gene that's being encoded, is transcriptomics, And there are about 100,000 transcripts. So as you can see, the level of complexity just increases as we sort of move down along this transition. And then the protein, which is the functional unit, is a study of proteomics when you look at them all together, and there are about 1 million proteins in the human body. So we want to understand the changes that occur at each stage. So there are several mechanisms that can lead to cellular abnormalities. There can be an incorrect copy number of genes. We acquire one copy from our dad and one copy from our mom, so we should have 2 copies of a functioning gene. We can either gain more copies of a cancer-causing gene, for example, or lose copies of genes that are important in tumor suppression. So if that happens, then the wrong gene, so to speak, we can lead to problems in abnormal cellular growth. We can also have improper gene regulation, which often happens, and this leads to the improper expression of a gene. And just to give you an example of how that is, when we eat, we turn on enzymes in our body to digest our food, but we don't want these enzymes to be continuously on. After we've digested our food, we want these enzymes to be turned off again, so we're not expressing these genes abnormally. Well, those control mechanisms are lost in cancer. We often have mutations in important genes that can lead to cancer, and we also can have abnormal protein function even in the absence of any of these abnormalities. So you can have a number of these occurring at the same time. You can have different, any combination of this can go on in a cancer cell. So some of the technology that we use, I'm just going to give you an example. So for DNA copy number changes, you have, this is how we pictorially look at DNA at losses or gains in genes. And so these blips to the left over here are an indication that we've lost that part of the genome. And there may be genes involved at each of these points that are very important to cancer. And so here are samples from different patients, and we can see that there is a region here that's pretty consistent across these samples, which tells us that the gene that's involved in this location could be very important to cancer because it's occurring in multiple patients. We can also have lots of structural abnormalities, and this is, even though it is an old technology that's used, it's just a good representation of what could happen. So here we have 2 chromosomes for every chromosome, so 1, 2, 3 through 22, and then the X chromosome and the Y chromosome if it's a male. But you can see here in a cancerous cell, you have all kinds of rearrangements, gross structural rearrangements of these chromosomes, and you have increases in the number of chromosomes, which obviously would also lead to increases in the number of the genes that are involved on those chromosomes. So a lot of things can really, really happen that just make a cancer cell behave in a very, very abnormal way. So this is an example of what it, just a diagrammatic representation of gene expression. We want to be able to express a gene and keep it off when we want and keep it on when we want, but we need to be able to control how genes come on and off. If we lose that control, like I was saying, we can lead to a lot of problems in the cell. An example of what happens or what could be the consequences of a mutation in a gene. So all these bases that make up the DNA are codes for various things in the genome. And so one of these codes could be a stop code to say, stop making protein now, all right? Because remember I said DNA goes to RNA, goes to protein. So the code is basically how the body knows to do that. Well, if there is a mutation in the wrong part of the code, it could lead to a premature stop signal. And it can make that protein much smaller and lose its function. And so that could be one of the consequences of a mutation in the gene. This is an example of how we could identify what proteins do or how they impact cells. So these are pictures of brain tumor cells stained for different proteins. So we can actually paint the cells with the proteins that we're interested in looking at. And we can also study the morphology of these cells, which is also an indication of how normal a cell is. And so this is just an example of a microscopic image of different brain tumor cells stained with different proteins that have different functions in the body. Now, as I had mentioned, I talked about the interaction. So these proteins don't work in isolation. They communicate with each other. And this is just sort of on purpose a very complex yet still simplistic representation of what a particular protein, any one of them, so you can see the protein ROCK right there in the middle, and how it interacts with all the proteins either above it in the axis of if it's signaling context, or below it, or laterally as well. So there are so many important things that have to happen correctly in a cell in order for a cell to maintain its normal function. And even as I said, the mutations aren't there, the copy numbers are correct. If this interaction is thrown off for whatever reason, it could still lead to abnormal cell function. At the end of the day, one of the most important things really is how a protein function is altered. That's the bottom line at the end of the day. So I've showed you some pictures to describe how we visualize, or how to, just to give you some analogies of types of genomics assays that we do. One of the revolutionary ways of doing genomics research basically had to do with technology referred to as microarray technology. And this is an arrayed series of thousands of microscopic spots of oligonucleotides, which are pieces of DNA, and these are called features. They each contain very, very small amounts of specific DNA sequences known as probes. And if we can interrogate a human DNA sample or an RNA sample with these microarrays. And some of the things that have come out of this is that we're now able to study not just a single gene at a time, which is the study of genetics perhaps, but many, many genes at a time. And so we can then look at multiple human samples across thousands of genes in one experiment, and we can begin to group patients according to the way they behave at the genomic level. And so you can see the lines, which are colored in blue or gray or cyan or magenta, red, and green. And those basically are samples that have clustered together because of the genomics assay that was conducted to say, these breast cancers look very similar to each other. These breast cancers look very similar. And so now we went from having or referring to breast cancer as one type of disease to actually it being at least 5 or 6 different types of diseases. So I said there are 100 different types of cancer, but even within each type, there are still multiple subtypes. And they can have different clinical manifestations and clinical outcomes. And so in order to better treat patients, we need to better understand how these breast cancers look at the molecular and genetic level in order to be smarter about the different treatments. So every breast cancer patient should not be treated the same way. These are the platforms, and this is what they look like. So we have, and this is just to give you an idea of what these machines look like. They actually rest, they're not that big, then they rest on bench tops. This is what a gene chip looks like. So this is what the actual microarray platform could look like. One of these things can basically get inserted into a machine that will scan it. It will give us a picture that looks very much like that. We have moved now to the next generation of genomics technology, and it's referred to as next generation sequencing. So the Human Genome Project took 13 years, $1 billion approximately, and an army of people to discover and unfold the sequence of the human genome. Well, now we can do that for thousands of dollars. It can happen in about less than a month with the analysis time, and it doesn't take a whole lot of people. So we've really moved to the next level, which is another revolutionary step in genomics research. And the other amazing thing about next generation technology is it combines a lot of those individual technologies to study the copy number changes, the gene expression changes, the mutational changes, and you can perform all of those readings on one machine during one experiment. So it's quite amazing. And then you can get these representations of and integrated representations of the compendium of changes in a cancer cell, and that circle that you see and all the layers of the circles that you see are basically a representation of all the changes that have occurred at each genomic level in a given tumor. And so we're using these types of technologies now to treat patients because we can learn about an individual's changes as opposed to figure out, as opposed to grouping people in these large, broader groups, we can treat people on an individual level. So we can also go from using single gene predictive tests to multi-gene predictive tests because of genomics, which has been useful in various aspects, and things like MammaPrint, Oncotype DX tests have been the result of some of those experiments. These were early on consequences or outcomes of some of the genomics research. So I'm going to switch gears a little bit and talk to you about the treatment plan and what it may include. It obviously could include surgery, chemotherapy, radiation therapy, hormonal therapy, biological therapy, clinical and clinical trials. So local treatment can be either local or systemic. Local treatments affect cancer cells in the tumor and the area near it, so it's very specific and localized to the area that's affected. And systemic treatment travels through the bloodstream, reaching cancer cells all over the body, and can often lead to not very nice side effects. And the side effects result from the damage to healthy cells and tissues in addition to the cancer cells. So that's why side effects, people have some of these very strong reactions to the various drugs available, because unfortunately, although it preferentially is supposed to affect just the cancer cells, it can often also affect the normal cells as well and lead to various side effects and consequences of that. So surgery, just to give you a brief description of each of the different treatment options, is the removal of the cancerous tumor and possibly the surrounding tissue, lymph nodes near the tumor. So surgeons always want to take as much of the tumor as they can. In some cases where microscopic cells have infiltrated the normal tissue, they may have to go beyond the margin of the tumor and just try to get as much of it as possible in order to remove some of these microscopic things, cells that may be present that aren't visual, surgeons are not able to see. Chemotherapy is the use of drugs to kill cancer cells. Biological therapies help the body's natural immune system to fight disease or protect the body from the side effects of cancer treatment. Radiation therapy is the use of high energy rays to kill cancer cells or stop them from growing and dividing. Hormonal therapy is used against certain cancers, such as prostate cancer or breast cancer, and depend on the hormones that are required for growth. Targeted therapy, which is some of the types of therapies that we all achieve for because they're more specific, they're supposed to be less toxic, but it's a type of medication that blocks the growth of cancer cells by interfering with a specific targeted molecule like a cancer-causing gene needed for tumor growth. And it bypasses just killing growing cells in the body. So chemotherapy targets growing cells. So cells of our stomach, for example, are constantly growing. And so people can, feel very sick to the stomach after chemotherapy because it's affecting the growing cells in the lining of the stomach. So targeted therapy is supposed to be much more targeted and specific. An example is Herceptin, which targets the HER2 oncogene in breast cancer. So today we have what we can say, refer to as a trial and error method of medicine. We may have a good idea of who, or we may be able to predict to some degree who will respond to a drug or not, but we really can't. We only still have about a 30 to 60% response rate to drugs. And what happens to everybody else that doesn't respond? We have to try other things. And so with personalized medicine, what we really want to achieve is a 100% response rate. We want to target the cancer in a specific person. So we want the right treatment for the right person at the right time so that we can have a 100% response rate. And personalized medicine, just to give you a little bit of definition on that, is a method in which diagnosis and treatment of a medical condition is catered toward that particular patient's genetic profile to compensate for metabolic differences, mutations that aren't known, inactive viruses that may be playing a role. So there are so many things that can go into why a patient responds or does not respond. And we need to better understand these differences so that we can achieve that 100% response rate. And so what happens is that if you look at this graph, with the current trial and error method, if the first diagnosis and treatment plan doesn't work, it may be too late to save the patient, but because by the time a physician comes up with another thing, the disease may have already progressed to a point where the symptom severity has increased to a point where disease management just falls off the cliff and becomes harder and harder to achieve. So we really need to do a better job, and we're really working hard on this. So again, just to emphasize, the goals of personalized medicine are to identify genetic differences between people that affect drug response, to develop genetic tests that predict an individual's response to a drug, and to tailor medical treatments to the individual to increase effectiveness and, of course, more importantly, or just as importantly, minimize these adverse side effects. So the old way is to take a patient's tissue sample, look at it under the microscope, make a diagnosis, come up with a treatment decision. But the new way is to take a patient's tissue sample or blood sample, perform genomic analyses on the tissue sample, whatever technologies may be available, and to cater treatment not just based on pathology that the pathologist sees, but also based on the genetics of that individual patient. So this is just a list for you to look at. I won't go through it in a lot of detail about some of the advantages of personalized medicine. Of course, it's going to reduce the time, cost, and failure rate of medicine and clinical trials. It's going to avoid withdrawal of marketed drugs. It may actually reintroduce drugs that may have been removed, because even though it didn't work for the majority of people, it may work for some people, and so that would be still a good thing, and those people should benefit from those drugs. So there are a lot of advantages to really moving towards a personalized medicine that's based on or driven by genomics research. All right, so everything that I've been saying really has been talking about or referring to translational research. And translational research is a branch of medical research that attempts to more directly connect the basic research to patient care. So the very nitty-gritty of science to patient care. How do we take that information that we learn at the bench in the lab to the patient bedside? All right, and so it's any form of research aimed at generating data which could have potential diagnostic, prognostic, or therapeutic implication. So in other words, it's a bench-to-bedside approach. And the very first thing that has to happen often is a discovery needs to be made. All right? That discovery has to be validated. So just because we make a discovery, it doesn't mean it's going to lead to anything. We may discover that there's a loss of a gene that seems to be very important. It seems to be a frequent thing that's happening, but we need to validate it. And there are ways to validate this. We need to make educated guesses about what we think that genetic alteration is doing to the cell. We can design and develop models both in the lab and in vivo, so in animal studies, to explain and better understand the function of this genetic alteration. And we can also establish and need to establish clinical significance in order to move it forward. So is this discovery going to be clinically meaningful? This is the bench component of the translational research process. Clinical feasibility involves drug discovery, it involves development of maybe diagnostic assays, it involves integration and mobilization of academia and industry. We need physicians, we need scientists, we need business development people all to be involved in this process. And we need to then really start to think about clinical trials if we are so lucky to reach this point. This is the bedside component. So essentially what we're doing is we're studying, we're analyzing the tissue of patients, we're performing the genomic analyses, and then we're going back to the patient with the proper treatment based on that. Now, this is not that straightforward in the sense that it takes a long time and a lot of money, and the success rate, unfortunately, is not as good as we would like it, although with the advent of all these different types of genomic technologies, particularly next generation sequencing, hopefully we can reduce these costs, maybe not costs right away, but we can reduce at least the time. So for every 5,000 to 10,000 compounds that are screened, so in other words, you could say for every 5,000 to 10,000 discoveries, 5 of those may actually make it into clinical trials. And this takes about 14 to 16 years of research and about $1 billion. So it's a long time, it's very expensive, but it's how a lot of things, it's how everything, all the drugs that are being used today have come to be used. So what is a clinical trial? A clinical trial is one of the final stages of a long and careful research process like I just described. Studies are done with patients to find out whether promising approaches to prevention, diagnosis, and treatment are safe and effective. There are different types of clinical trials. There are treatment trials, which test new treatments like cancer drugs and new approaches to surgery or radiation, combination treatments, or new methods such as gene therapy. There are prevention trials that test new approaches such as medicines, vitamins, minerals, or supplements. These trials look for the best way to prevent cancer in people who have never had cancer. So that's one type of trial. Another trial is a screening trial, and this is to test the best way to find cancer, especially in its early stages. So early detection, we all know, is very, very important to a better outcome of disease. So constantly working on projects that allow us to improve the screening for different cancers is very important. We have very good screening approaches for some cancers and none or not very effective ones for others. So we need to improve that. There are also quality of life trials, which explore ways to improve the comfort and quality of life for cancer patients, which is equally important as all the other types of aspects to the cancer battle. This is just here for your own purposes. You can read this on your own time, and it goes into a little bit more detail about the different phases of clinical trial. There are 4 phases of a clinical trial. So after the preclinical phase, which is the stuff that happens that the scientists do at the bench, they may have proven feasibility in an animal model. It then, with the right combination of events, it can move into a clinical phase, and then there are 4 phases to the clinical phase before it gets approved by, or in the last phase, it's approval by the FDA and then used as hopefully new standard of care. So this is just to show you how far we've come over the last, I don't know, few decades. We used to just treat cancer by, just removing entire organs. Then the advent of radiation and chemotherapy made a huge difference in the success of how people do. But then we started to become a little smarter, and we started to understand that targeted therapy may actually be a better way to go, and that was because we started to understand the function of genes and learn about what are the different genetic alterations that drive a particular cancer. And so the scientific field has achieved things like the tamoxifen and things that block things like the estrogen receptor. And I'm just giving you an example for breast cancer. But we're moving from these single gene targeted approaches, which are still very important, will still be used, to these multi-gene ways and comprehensive and integrated ways of understanding cancer and trying to target it. I want to talk to you a little bit about the clinical trials that happen at TGen, which we have. TGen's Clinical Research Service is a large phase 1 clinical trial center which was launched in 2005. And these clinical trials give options that did not exist before to the Phoenix area patients, as well as patients from all over the country. They're involved in clinical trials across a number of different cancer types, such as pancreatic cancer, the big one, breast cancer, leukemia, prostate cancer, lung cancer. In addition to clinical trials, they performed one of the first molecular profiling pilot studies that have been designed and implemented to test the hypothesis that the treatment of a cancer with a specific agent is based on the unique molecular profile of an individual patient. And so this has shown some great efficacy and was a pioneering study in the field of personalized medicine. This is some information that you can use. This is contact information if you want to be able to contact TGen and explore clinical trials options. Clinical trial enrollment overall across the country is actually very poor, but I encourage you to always consider it and keep that as an option in your care if need be the case. If obviously you're doing well with your current treatment and everything's going fine, then you may not need it. But if you're looking for other options, I would encourage you to consider clinical trials. Here are some useful websites. The first two are websites for clinical trials. You can search clinical trials across the country, in fact, North America, Canada, and the US in particular. And they have very user-friendly websites that you can use to look for ongoing open active trials either in your area or across the country. And some other cancer organization websites. There are numerous, but these are just ones that I think might be good. The American Cancer Society, the NIH, NCI, the Center for Disease Control, and Komen, only because it's really an excellent website. And that's all I have. Thank you very much.